Hey again everybody, here we are talking about action potentials. Now if you remember last lecture we went over our graded potentials and we talked about resting membrane potentials. So today we're going to look at what that action potential is. So just to recap, we talked about neuron structures, we talked about current and electricity, we talked about ions. Last lecture we talked about how that resting membrane potential is maintained. And then we talked about what happens at that membrane when some kind of stimulus, either an excitatory or inhibitory stimulus, comes in, and how that brings us closer to or farther away from our threshold. Today we're going to start at that threshold and we're going to look to see what happens when we hit threshold, how we get that big blip and big change in voltage, and how that's transmitted down our axons. So start here. How is our resting membrane potential maintained? What's the difference between a graded and action potential, and what type of channels would be involved? All right, so how to go? If you said our resting membrane potential is maintained by leak channels, you would be correct. If you said the sodium-potassium pump also helps with this, you would be correct. Remember that sodium-potassium pump is expending, especially in neurons, about 70% of that cell's energy just to maintain those concentration gradients of sodium and potassium. What's the difference between a graded and an action potential? Well, graded potentials are decremental. They get weaker over distance. They can stop or be reversed. They can occur in the dendrites or in the cell bodies, and they can be both excitatory or inhibitory. It just depends on which membrane proteins are opened and which ions are moving in. Are positive ions moving out? Are positive ions moving in? Are negative ions moving in? So we want to really just kind of know more about our proteins to see which ions are moving. Action potentials then, on the other hand, are all or nothing. They're not reversible, and they are excitatory, and they occur along those axons. And then finally, what types of channels would be involved? And so hopefully you said ligand-gated channels when neurotransmitters are going to bind, and voltage-gated channels are going to open during an action potential. Now we can also, if you remember, said that during touch receptors, we have mechanically gated channels that may or may not open. So basically all three of our channel proteins. Now, we got to this point last time and we said that local potentials are coming in from a presynaptic neuron. Some kind of stimulus is coming in, and that's depolarizing our membrane. But that depolarization has to reach negative 55 millivolts. It has to reach our threshold. Otherwise, we don't get a signal that is sent. So you can see here, these are these failed depolarizations. But we can see here that this is going to take off. So threshold, in our case, we're going to use negative 55 millivolts. As you'll learn down, in your academic careers, you'll see that the resting membrane potential and threshold for different cells can be different in our bodies. But for our purposes, for these lectures, negative 70 and negative 55 are the two that we'll be talking about. Our minimum has to be hit in order to open voltage-gated sodium channels. Let's start here. You're going to be pretty interactive with this lecture. So what I want you to do is pause the video, and I want you to draw a neuron, and I want you to draw two neurons. Actually, let's start there. Let's have you draw two neurons kind of connecting with each other. And then below it, what we want to do is I want you to draw a neuron membrane, and I want you to label which side on your drawing is intracellular and which one is extracellular. And I want you to put down the voltage. Is it a positive on the inside or outside? And where is negative? So go ahead and draw that first, and then we'll move on. All right, you got it? So what we're going to do is we move forward and we talk about each of these phases or stages you're going to go back to your drawing and you're going to add in some information and you're going to change things around, or you can put arrows in there, okay? But we want to try to apply this as we go through this lecture. So here's maybe what you have. Hopefully you drew that phospholipid bilayer, and you've got our hydrophilic heads and our hydrophobic tails on the inside. And you may have said the intracellular space is down here and the extracellular space is out here. And you should have said that the inside of this membrane is more negative and the outside is more positive than it is on the inside. And remember, it's a relative measurement, and so we have a negative 70 millivolts, and that is going to be our resting membrane potential. All right, now let's talk about this depolarization. So when we get excitatory signals that are coming into that postsynaptic cell, we are getting those local potentials, and if they're large enough or long enough and big enough and have an additive effect that brings us to threshold here, what is going to happen? Well, once we hit threshold, those voltage-gated sodium channels open really quick along that membrane. At the same time, slow potassium channels start opening, but again, they're real slow. So if you have voltage-gated sodium channels opening really quickly and you have some slow potassium opening, remember, sodium is going to rush into the cell down its concentration gradient, and you get some of those positive potassiums moving out, but much more slowly. So overall, we're increasing the positivity of our membrane here. So sodium enters, and as it's entering, it's causing more voltage-gated sodium channels to open. So we get this massive influx, really quick depolarization. Remember, moving away from our resting membrane potential and becoming more positive. So what you're going to do now is add some gates and some ions into your drawing. Where do those sodium and potassium gates exist, and draw in those ions at this point during a depolarization. So what does your neuron membrane look like at this point? Okay, so hopefully that wasn't confusing, and what you should have added to your drawings is that you have open sodium channels. So sodium is rushing in, as we said, stimulating more sodium channels to open and causing a quick increase in that voltage. You should have also drawn in some potassium channels that are kind of opening real slowly. They're taking their time. They're a little bit, harder to wake up in the morning, and things are moving through them much more slowly than this sodium. Sodium is rushing in real quick. So what happens then is our voltage eventually peaks. And so when our voltage passes 0, our sodium channels start becoming inactivated and they start to close. So again, when you look at that sodium channel, negative 55 is going to open it up. At 0, they're starting to close. So now, all these sodium channels are slamming shut, so no more sodium is coming in. And by the time it reaches positive 35, they're all closed. Okay, so now we've got 4 voltages to know about. Negative 70 is our RMP, negative 55 is our threshold, 0 causes our sodium channels to close, and at 35, those sodium channels have all closed. Now, if you remember, those potassium channels are still open. okay? And they were those slow potassium channels. So potassium is now going to be diffusing out. Now our membrane is more positive on the inside and more negative on the outside, okay? Because all of that sodium rushed into the cell. So that's the opposite of our resting membrane potential. So again, pause here, draw in what your membrane looks like when the voltage has peaked. So where are the concentrations of ions? Which side is positive? Which side is negative? What do your ion channels look like? All right, so how'd that go for you? Hopefully what you put in there is that at our peak, our sodium channels are closed. We have a lot of sodium inside of our cell, making it much more positive, positive 35. And sodium is now rushing out down its concentration gradient, but still much more sodium has come in than potassium is going out. So it's more negative on the outside, more positive on the inside. But as you can see by this graph, we're starting to repolarize. So we're starting to return back to our resting membrane potential. Now, if we continue to look at repolarization, our potassium channels are open. So potassium is diffusing out of the cell, and that is going to return that voltage to a more negative state, and it's shifting back towards its resting membrane potential, and we call that repolarization. So now, go ahead and draw again, or add to your drawing. What does it look like after our voltage peaks and it's starting to repolarize? And don't forget, our leak channels and our sodium potassium pump are also present. So are they working? Okay, now what you're going to see here is that as we're going through this repolarization process, those sodium channels are still open. Sodium is moving out, and our sodium potassium pump is still working, and so are the leak channels. So we're still having sodium and potassium diffusing. We're still having sodium and potassium being pumped in and out with our sodium potassium pump. So hyperpolarization then is our last stage, and those K channels, those potassium channels, are, remember, slow. And so if I were to, if you've ever seen a door that's kind of an auto door that opens and closes, you hit the button, the door opens, and then it closes, and it's kind of slow. And if there's no auto sensor, people could still kind of run in through that door as it's closing, and then eventually it closes and slams shut. This is kind of the same idea here, is that there's a delay. Those potassium channels take their time to fully close, and while they're doing that, the inside of the cell is losing potassium outside of the cell. And we get this little hyperpolarization, or a move that becomes a little bit more negative than the original resting membrane potential. Then after those potassium channels have fully closed, again, our leak channels and our sodium potassium pump are going to get that resting membrane potential brought back to negative 70 millivolts. Now, we also have astrocytes. If you remember our glial cells, they are helping remove extracellular potassium along with that sodium potassium pump and those leak channels. Okay. So really quick to point out that most often when we study action potentials, we see a graph that looks like this, and we see time. Okay. But what it actually looks like is much, much more rapidly than what it's presented in a graph. We spread it out so we get an understanding of the different stages, and we can talk about the different channels that are opening and closing along the way. Here is our stimulus that gets us to threshold. Here's our action potential, this spike, and back to our resting membrane potential in about 50 milliseconds. So this spike is actually only happens in a handful of several milliseconds. Our hyperpolarization then is a little bit longer, much longer than this spike here, and we're going to talk about why that's important. But again, we're only talking about milliseconds here, so this is a very, very quick process. So practice first, and what I'd like you to do is to draw a graph of an action potential, so kind of like what we just looked at, millivolts versus time, and I want you to label the following regions. So where's our resting membrane potential, our local, our action potentials, depolarization, repolarization, and hyperpolarization. Try to do it without going back to the previous slides. See how well you remember this. Force your brain to fire its action, or fire its neurons to think about what we just went over. All right, so how to go. Hopefully you were able to apply some of that, and what you didn't figure out or what you couldn't remember, you were able to look up and figure out and apply that. So again, practice this. Draw it and redraw it and draw it and redraw it and draw yourself a couple of neurons and talk through this process of here's a stimulus, we have an EPSP, we have an increase in our voltage, we hit threshold, and what's happening along the way. Talk about the ion channels and bring it back to resting membrane potential. Do it over and over. This is tricky stuff. Now another way to just look at this, I kind of like this diagram. So you can see down here we have our resting potential, and so here's inside of our cell, here's outside of the cell. You can see we have a higher concentration of potassium, higher concentration of sodium out here, and you can see the voltage that we have. During an action potential, we have depolarization, so we can see what's happening with our ions, we can see what's happening with our voltage. During repolarization, you can see what's happening with our ions and our voltage. We're returning back to negative, and then again at resting potential, we see that sodium-potassium pump working, and maybe those astrocytes helping, remove that extracellular potassium. So again, it's just a different way to reference this, and I would keep that on hand. Maybe that'll help you study this a little bit more. Okay, so here's your favorite diagram. Know that you will see this diagram, this exact diagram on a test. You will not have labels, but there will be numbers here, okay? And I'll be asking you what's happening to the ion channels, what's happening, is it a repolarization, is it depolarization? I might ask you true-false statements and say, which of the following statements are false about this diagram, and give you, 4 true statements and 1 false, or, vice versa. So, all right, now, we left off talking about our hyperpolarization, and we said that it actually is a little bit longer than that real quick spike, which is only several milliseconds, and that hyperpolarization is actually maybe 30 or 40 milliseconds. So we have something that's called a refractory period, and this is really important in making sure that that postsynaptic cell isn't firing again. So during that action potential and a little bit after, it's very difficult or impossible to stimulate that part of a neuron. Okay, and so we have 2 types of refractory periods, and a refractory period is a period or a time of resistance to stimulation. Resistance to stimulation is key when we're talking about these 2. So here we can see we've got green, good to go, we've got red, not going to fire, and we have yellow, which is caution, we might fire. So we can term those absolute and relative refractory period. So there's 2 phases. That absolute refractory period means that no stimulus will actually trigger another action potential. Okay, and that is the part of the membrane that is actually experiencing an action potential already. So any other stimulus cannot stimulate this part of that axon. The relative refractory period then is you can still stimulate another action potential, but you need a much stronger stimulus because, remember, our membrane potential is more negative than the resting membrane potential. So now, if we have a difference of 15 millivolts here between our resting membrane potential and our threshold, but if we have the hyperpolarization or inhibition, and now we're at negative 75 millivolts, we have a difference of 20 millivolts. So it's going to be a little bit harder to get back to threshold. And so we need a bigger stimulus in this little relative refractory period. Once our cell membrane returns to resting membrane potential, now it's ready to fire. We're not in a refractory period. So this is because if we're talking about that relative refractory period, it's because those potassium gates are open and any chance of that sodium coming in is kind of washing out or offsetting the sodium coming in by the potassium leaving. So let's look a little bit closer. If we look at the refractory period, okay, and we take this chunk of our axon and we blow it up, we have right here, we can see that sodium is rushing in, and we've changed our voltage here on this membrane, where down our axon we don't have anything changing, right? Nothing has happened yet. But what's happening is as that action potential is moving down our axon, because remember, our neurons are communicating in one way, from the dendrites to the axon terminals. So sodium moves in, it stimulates more sodium channels to open, and those sodium channels open, which stimulate more sodium channels to open, and that action potential is moving down our axon. But behind it, remember, our potassium channels are slower to close, so potassium is rushing out, making it more negative and more negative than that resting membrane potential. But eventually, RMP is returned, so resting membrane potential is returned, and we could have another stimulus back here. So now, on your graph, draw in where your absolute refractory period and your relative refractory periods would be. Okay, so hopefully you did that, and that was pretty easy for you. But what I would encourage you to do is to also go back and revisit that graph and annotate a little bit about what is an absolute refractory period and what is the relative refractory period. Okay, so a little bit more. Moving on, when we talk about our action potentials, it is always occurring on our axon. It's all or none. So once we hit that threshold, there's that trigger zone is fired and our action potential is sent down our axon. It's not decremental. Okay, so remember, with our graded potentials, we saw that it sort of dwindled. With our action potentials, we're hitting that spike up to a positive 35. It's not reversible, so it's not going to go back, and it can travel a really long distance in our bodies. Now, it has to get to the end of our axon, because what we'll see in our next lectures is as that signal is traveling down our axon, it has to reach those terminals. And in order for it to communicate chemically across the synapse, that action potential needs to stimulate those axon terminals. And if you remember when we talked about multiple sclerosis, if we have the degradation of those myelin sheaths and a replacement in scar tissue, that signal may not get there, or it may jump to another neuron. So what does that look like in our nerve fibers? Well, they're self-propagating, and so as long as there are voltage-gated ion channels, we get this action potential to be propagated down that axon. If for whatever reason there were no voltage-gated ion channels at this region of the axon, you wouldn't be able to open those channels because they're not there, and you would cease your signal. So unmyelinated axons, if you remember we talked about myelinated and unmyelinated axons, they have channels along the entire length. And if you remember, we said unmyelinated axons are a little bit slower, and we find these in our pupils, and we find these in our digestive system. So areas where we don't need these rapid signals sent to. So basically that action potential, you can kind of think of it as like a domino moving down the axon terminal, sodium coming in, triggering more sodium channels to open, coming in, triggering more sodium channels to come in, and so on. So why isn't that action potential then moving backwards? Well, if you look at this diagram, here's showing our action potential, and so right here, what we have are our open sodium channels. And so sodium is moving in, and you'll notice with this arrow on the right and this arrow on the right, that as that sodium is moving in and diffusing into the axon, it's triggering the cell, or the ion channels that are closed to open. So more sodium comes in, which triggers the opening of more channels, and more sodium, and more, and more, and more. But remember, behind it, we are repolarizing. So those sodium channels are closed, and they're also inactivated. So if sodium is coming in and diffusing back this direction, it's not triggering these channels to open. So there's no more sodium coming in, and what we're having happen during this repolarization and hyperpolarization phase is potassium moving out. After we get back to resting membrane potential, those channels are ready to be opened again. So this is how our neurons allow for or ensure that this signal is moving in a unidirectional manner, okay, and not moving backwards. So another way to look at this, and I like this visual because it's kind of showing us our action potential in progress, our refractory membrane, and our excitable membrane, so we can see the different voltages along this axon, and you can see how here is our action potential. It's moving down our axon. You can see our refractory period behind it, so these sodium channels aren't going to open, and this is going to follow in closely behind our action potential, and then we restore the excitability behind it. Now remember, in the red zone, this is our absolute refractory period, so there's no other stimulation that's going to do anything here because we're already in an action potential. In the yellow, we just need a lot stronger of a stimulus coming in to get us closer to threshold and fire. So it can happen, but that's our relative refractory period. If we look a little closer at those myelinated fibers, remember we talked about our myelin sheath, so our oligodendrocytes or our Schwann cells, depending on where we are. In between them, we have those nodes of Ranvier. So in myelinated nerve fibers, our voltage-gated channels are needed for these action potentials to occur. And so what we can see is that in these myelin-covered regions, also called internodes, we have voltage-gated channels, but they're a lot less dense. Whereas if we look at our nodes, they are very dense, and you can see how much difference there is between the two regions. Let's look a little bit closer at why. So if we look at our nodes of Ranvier, you can see that sodium flows through those voltage-gated channels. They're much, much more dense and much more present than in between our internodes here, where our myelin is. So sodium flows in at the node and generates an action potential. The sodium diffuses on the inside of that axon and moves across pretty quickly, and again is going to stimulate the opening of those sodium channels here. So it looks as though our signal is sort of jumping from node to node to node. and we get the term saltatory conduction, where that nerve is kind of jumping between those nodes. Whereas if we're looking at an unmyelinated nerve, that signal is being passed all the way down the entire length of that axon, and so there's much more surface area for it to cover. Whereas here it can kind of fire and jump, 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 and get to the axon terminal. So again, just to kind of reinforce the color schemes that we had talked about before, and we add in now our myelin sheath, we can see here's where our action potential is. We have some diffusion through here, which triggers the opening of sodium channels here, high density of sodium channels. That diffuses and rushes through, and we get opening here, rushes through, opening, opening, and then you can see behind it is where we're repolarizing and hyperpolarizing, resetting those ion channels, and eventually our membrane is ready to go again. So if you remember these videos, pause your video here and think about what neurons have to do with flushing a toilet and shooting a gun. All right, welcome back. Now I hope that that's fairly obvious as to what's going on here and how these are similar. If we talk about a toilet, for example, we have a full toilet that is our resting potential. It's ready to go. When we reach threshold, we push the flush lever, right? You push on the lever a little bit and you hear it move, but nothing really happens. If we push it all the way down, that water rushes out. And so this is our action potential. The toilet then is refilling, and this is our repolarization. You're trying to push the handle down because whatever you put in there doesn't all go down. You're like, oh man, come on, right? And you have to kind of sit there and wait for the water to get to a certain level. Now, we've all done it where, you wait, you're getting impatient, and you push it, and it's not enough water because you haven't let it fill all the way up. That's your relative refractory period, right? You get a little bit of a signal, but you have to wait until it refills all the way. And then our sewer and our drain pipes is that one-way communication, right? Unless you're, doing something else and putting some things in there that shouldn't be in there, you don't get a backup. But that communication goes one way, goes from your toilet out the drain, out into the sewer, and it doesn't come back, hopefully. Again, just to touch on myelination, the size of the neurons can affect that conduction speed. So the larger the fibers are, and we talked about this before, the faster their signals are going to go than those smaller fibers. And because there's less resistance, so remember, sodium is coming in and it has to diffuse down that axon. And so if you have a larger diameter of an axon, those sodium ions move along much faster, and there's much less stuff in the way, right? Where here, if you have a thinner diameter or smaller diameter, you can see some of those neurons are running into other organelles or molecules inside of that cell. So our speed is going to go faster, and there's much more area in this one than in those smaller ones. Well, folks, that is the end of our action potential lecture. Here's a couple of questions to just think about as you move on. When we come back for our next lecture, lectures 15 and 16, we're going to be looking at neurotransmitters, how they cross our synapse, and we're going to look at some specific types of neurotransmitters and what effects they have on our neurons. So good luck studying. Study well, study hard, study long. See you next time.